Hello, so we are going to start alternative to practical today again, like yesterday. So obviously you can find this in your syllabus, paper 6. So let's look at this together because this is an important part to read. Alright, so the alternative to practical is designed to test candidates' abilities to follow carefully a sequence of instructions. So obviously all the instructions will be given to you in the question to describe familiar and suggest unfamiliar techniques, to record observations and make deductions from them. Now, to recognize and observe features of photographs, so you also need to be able to identify photographs and also draw them, familiar and unfamiliar biological specimens, to record their observations and make deductions about functions of whole specimens or their parts, to make clear line drawings of the images of specimens, indicate magnification, and label familiar structures, okay? To interpret unfamiliar data and draw conclusions from their interpretation. So you need to be able to draw conclusions, basically. To design or plan an investigation, to solve a problem, to comment on a procedure used in an experiment and suggest an improvement. So yeah, you need to be able to comment on procedures and experiments and also suggest improvements. Okay, so basically what are the instructions and experiments, you need to be able to understand what they're trying to do, and also realize that probably that is not the best way to do such an experiment. So you need to think of improvements as well, or some errors, maybe possible errors. To observe images of reactions, read from photographs, diagrams, and simple measuring instruments, and perform simple arithmetical calculations. So yeah, to measure to an accuracy of 1 millimeter, so you have to use a ruler. Candidates may be asked to describe simple physiological experiments involving tests for food substances. So that is one. Enzyme reactions, hydrogen carbonate indicator solution, so you'll be able to see in the papers, cobalt chloride paper, and other materials listed in the syllabus. Now, it is expected that candidates will have experience of the use of glassware and instruments normally found in the laboratory. For example, beakers, test tube racks, funnels, thermometers, droppers, and other apertures, okay, so that they can describe their use in such experiments. And candidates may be asked to describe simple physiological experiments involving the use of the items mentioned above in 1H. So, for example, this one. So, involving plant or animal materials. Accurate observations of life-sized and magnified images of such specimens will be expected. Okay. Now, the material set will be closely related to the subject matter of the syllabus, but will not necessarily be limited to the particular types mentioned in it. So yeah, there will be some unfamiliar experiments like they mentioned over here on top. In order to assist their own practical work, schools are recommended to build up a reference collection of material. When planning practical work, teachers should make sure that they do not contravene any school, education authority, or government regulation. Okay. Now, main thing is that to actually prepare for alternative to practical, I recommend practicing basically all the past papers, and like you have actually seen in your physics alternative to practical paper, that you actually do not need to have much theoretical knowledge. Mainly the thing is you have to read and understand the question carefully, and maybe you need very simple knowledge of biology in this case, or in physics case. So that's it, and then you'll be able to answer everything properly. Okay, so basically read and understand the experiments, whatever is given to you. Okay, the rest is all practice. So yeah, let's get started. I think I have another page for you that actually lists down all the apparatus and the equipments that is used in the lab, and for paper 3, and also that will be handy for paper 6, because you will be actually you'll be asked to not draw but or name them, but you'll be reading them in the instructions as well, so it's better to know what those are. Okay, so for example, here I have this list that they've given. So for apparatus and material safety equipment appropriate to the work being planned, but at least including eye protection, such as safety spectacles or goggles. Okay, now you need to be able to recognize these chemical reagents. Okay, so let's start. So for hydrogen carbonate indicator, bicarbonate indicator, you'll be using these a lot. Iodine in potassium iodide solution, we all know where we use this. It's usually to test for starch. And then Benedict solution, the same thing. It's basically just iodine. I mean, it's also used for starch. Alternative for failings. Then we have burette reagent, or sodium or potassium hydroxide solution, and copper sulfate solution. You'll also be using these. Ethanol and methylated spirit. Yeah, ethanol will be used as well. Cobalt chloride paper, pH indicator paper or universal indicator solution, or pH probes, litmus paper, glucose, sodium chloride, aluminum foil, or black paper. This is just table salt and glucose. Everyone knows what a glucose is. And the instruments, you'll be using rulers, of course, which are able to measure millimeters. So yeah, the precision should be 1 millimeter. And mounted needles or seekers or long pins with large heads. I don't think you'll need them in paper 6, but still, okay. Means of cutting biological materials, for example, scalpels, razor blades or knives, scissors, forceps. Means of writing on glassware, for example, wax pencil, marker, adhesives, labels, and pencils. Okay, all right. That's that. You don't need to obviously use them. You just need to know what those are. Okay. Next, the glassware and other apertures, some of which may be glass, plastic, or metal. Beakers or other containers, test tubes, test tube racks, and test tube holders. Funnels, droppers, or teat pipettes, or plastic or glass dispensing bottles, dishes such as petri dishes. Obviously, these are all glasswares and containers. So just gonna tell you, funnels is just basically something that looks like this. Funnels, it's just basically used so that you can pour liquid. Droppers are basically just teeny tiny droppers to just add maybe a drop or something. Dishes or petri dishes are basically where bacteria culture or any microorganisms are grown. Means of measuring small and larger volumes, such as syringes, graduated pipettes, or measuring cylinders. Syringe, we all know what a syringe is, and graduated pipettes. Pipettes are something like this, a very thin glassware, which is used to actually transfer liquid. So it is just used to transfer liquid. Okay, measuring cylinders, we all know what measuring cylinders are. It looks like this, and It has scales on it, so you can measure how much liquid, the volume of liquid you are, for example, transferring or whatever. Glass rods are just rods that are used to stir. Capillary tubes are basically very thin tubes. Okay. Glass slides and cover slips, these are basically used for microscopic slides to prepare samples for microscopic observation. So that's where we use glass slides and cover slips on top of that basically sample. So that is the cover slip, and that is, let's say, the glass slide. Okay. Thermometers covering at least the range of 0 to 100 degrees Celsius. Means of heating such as Bunsen burner or other gas burner or spirit burner. Wide tile or other suitable cutting surface. Whisking tube or other selectively permeable membrane material. And basically hand lens. Okay. That's that. You don't really need all of them. I'm just teaching you to be familiar with all these glasswares and apertures that you might come across in the lab or even in paper 6. Yeah, did I mention all these tests? I think I have. Like I said, iodine and Benedict solution mainly used for starch test. Biuret reagent mainly used for peptide bond. Yeah, test for peptide bond ki amra shobai jani, hopefully. These are bonds found in proteins. Okay. And then that's it, right? Ethanol, methylated spirit, these are just, I think, used to clean the different glasswares. But yeah, pH indicator, paper, universal indicator. We all know we need those to obviously measure our pH. What else? Okay, by the way, this is another type of pH indicator, this one. That's why it's there. I didn't mention that. Hydrogen carbonate indicator or bicarbonate indicator, that is basically another type of pH indicator which is very sensitive. Okay. Very sensitive. It can show you a range of colors and for a range of pH. Okay, that's it. Now let's actually start a paper. Okay.